Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. If you like this video, subscribe my channel and click on bell icon to get notification. In this video, I am going to explain one of the topic in transport in plants. It is mechanism of transpiration, various experiments related to transpiration and its significance. So water is transient in plants that can be movable from root to stem, stem to leaf, leaf to surrounding atmosphere. At this time, less than 1% of water it reach the leaves, and this water can be utilized by the plant for photosynthesis and plant growth. So most of the water is lost through stomata from leaves, and this loss is known as transpiration. So transpiration is the loss of water in the form of vapors from the aerial parts of plant body. In the structure of stomata, so transpiration is the evaporative loss of water by plants, and it is mainly associated with stomata present in leaves. And stomata is the main structure to perform most of the transpiration, nearly 50 to 97%. In most of the plants, stomata are open during daytime and closed during nighttime. Such stomata are called photoactive stomata. But in case of xerophytic plants like camp plants, stomata are open during nighttime and closed during daytime. Such stomata are called scato-active stomata. The opening or closing of stomata. The process is completely depends on structure of stomata and change in turgidity of guard cells. Stomata are the thin apparatus found in epidermis of leaf and young stem. It performs transpiration. So each stomata, it is surrounded by two specialized epidermal cells called guard cells, and these guard cells are differ from other epidermal cells due to presence of chloroplast and along with its structure and shape. The inner wall of the guard cell is thick and elastic, whereas the outer wall is thin. Due to turgidity increase within two guard cells. At this time, the two guard cells flanking each stomatal apparatus are pore, and the thin outer wall bulge out and force the inner wall into a crescent shape. When the guard cell gets crescent shape or half moon shape, immediately it promotes opening of stomata, and opening of stomata is also aided or supported by the orientation of microtubules present in the cell wall of guard cells. So these microtubules are made up of cellulose, and these are oriented radially rather than longitudinally, and it helps to open stomata easily. The guard cells are bordered or surrounded by one or more modified epidermal cells. These cells are called subsidiary cells or accessory cells. When the guard cells lose turgor pressure or turbidity due to loss of water, this elastic inner wall and reason their original shape, the guard cell become flaccid after getting its original shape, then that leads to close of stomata. So generally, the lower surface of dorsiventral or dicotyledon leaf, it consists of more number of stomata. But in case of isobilateral leaf or monocot leaf, it consists of equal number of stomata on both sides. In dicotyledon plants, the shape of the guard cell is either bean shape or kidney shape, but in case of monocots, it having dumbbell shaped guard cells. When you observe the mechanism of opening and closing of stomata, during closing and opening of stomata, the opening of stomata and closing of stomata, it is mainly depends on turgidity and flaccid state of God cells. During this subsidiary cells, it play a major role, and these are helpful for opening and closing of stomata to perform transpiration. Along with this, gases exchange mechanism take place for photosynthesis and respiration. The most famous theory to explain the opening and closing of stomata is active K plus transport or potassium pump theory. This theory was given by Leavitt in 1974, and according to him, he explained that the influx of K plus ions in God cells and their critical role in stomatal movement. The opening mechanism of stomata in light. During daytime, the starch is synthesized in God cells, and this starch is incompletely oxidized into phosphoenol pyruvate. Simply, it is called PEP. This phosphoenol pyruvate then it converts into malic acid by an enzyme called phosphoenol pyruvate carboxylase enzyme. This malic acid finally it is dissociated into malate ions, malate minus ions, and protons H plus ions in God cells. The H plus ions from God cells are transported to epidermal cells. At this time, to exchange, potassium ions are transported from epidermal cells or subsidiary cells to God cells, and it is carried out by some of the special pump proteins present in their plasma membrane are called hydrogen potassium ion pump proteins. In the God cells, K plus ions are balanced by malate anions. Along with these, some Cl minus ions are also absorbed into God cells, which neutralize some K plus ions, but movement of ions always required ATP energy. Such transportation is called active transport. So K plus and malate ions form potassium malate, and these potassium malate it is stored in vacuoles to maintain concentration gradient. So due to concentration gradient, the water present in surrounding subsidiary cells it enters into God cells to increase turgor pressure, and due to this turgor pressure, God cells are enlarged due to an osmosis of water that leads to opening of stomata. In the closing mechanism of stomata in dark, at night time, CO2 is not utilized in photosynthesis, so that the CO2 concentration increased in substomatal cavity. Due to this CO2 concentration, one of the plant hormone, it is called abscisic acid or ABA, functions to inhibit K plus ions uptake by changing the diffusion and permeability of God cells for positive ions. At this time, the K plus ions are transported back into epidermal cells or subsidiary cells from God cells that leads to decrease concentration and maintain concentration gradient. Due to this concentration gradient, water will move from God cells to surrounding subsidiary cells. Then the God cell gets flaccid state that leads to closing of stomata. The transpiration is affected by many external and internal factors. In this, the external factors it includes surrounding temperature, light, humidity, and wind speed. The temperature, higher temperature, it leads to more rate of transpiration but the transpiration decrease at low temperature. The light, more specifically blue light, it induces maximum opening of stomata. The relative humidity. So here the humid atmosphere, it leads to decrease transpiration, but when dry atmosphere is maintained around leaf, it promotes transpiration. Wind. Wind is not blowing and water vapors accumulate above the transpiring leaf, it decrease rate of transpiration. But when wind blowing, that can remove humidity and that increase transpiration. Availability of soil water. So the water present in soil, if it is not sufficient, that can reduce the transpiration rate. In case of high concentration of salt in soil also, water can reduce, that leads to reduce or less water absorption, that leads to less transpiration. Along with these various internal factors like number and distribution of stomata, along with the percentage of open stomata, water status of plants and canopy structure of plant, it regulate transpiration are considered as internal factors. The structure of leaf. Many features related to leaf structure like thick cuticle, waxy coating, thick-walled hypodermis, and sun-kissed stomata, it reduces rate of transpiration in plant. So these are the various external and internal factors related to transpiration. So to demonstrate the transpiration mechanism, the transpiration can easily demonstrated with the help of following experiments. In this, first experiment is bell jar experiment and second experiment is cobalt chloride paper test. In case of bell jar experiment, in this experiment, by placing a bell jar over a potted plant with pot is enclosed in a plastic bag to prevent water loss from soil, and after few minutes we can observe the drops of water it appear inside the bell jar or polythene cover that indicates transpiration. But in cobalt chloride paper experiment, the cobalt chloride, it is dissolved in water and filter papers are dipped and dried. After drying, it gets blue color. And when by keeping this blue color cobalt chloride papers on the surface of leaf by arranging with the slides, after few minutes, the blue color cobalt chloride paper, it turns into pink color when it touch water, that indicates transpiration occur from leaf surface. So the entire transpiration, it can be measured with the help of a device, it is called photometer. This photometer, it helps to measure the rate of transpiration in a plant. The significance of transpiration. So transpiration serve more than one purpose in a plant. In this transpiration can create or exerts transpiration pull for absorption and transport in plants, and this transpiration helps to supply water need for photosynthesis. Along with this, during transportation of minerals along with water from soil to various parts of plant body, it is due to transpiration only. Along with this, the entire plant body, more specifically leaf surface, it cools when water can transported through its structure, and it reduces up to 10 to 15 degrees centigrade temperature. Along with this, the transpiration, it helps to maintain the shape and structure of plant cell by keeping their cell in turgid condition. These are the significance of transpiration. So transpiration and photosynthesis, a compromise. An active photosynthesizing plant it need good quality of water and minerals. But photosynthesis is limited by availability of water which can be swiftly deplete by transpiration. The humidity of a rainforest is largely due to the vast cycling of water from root to leaf, leaf to atmosphere. Again, it came back to soil to maintain humid condition in rainforest. The evolution of C4 photosynthetic system is probably one of the strategy for maximizing availability of CO2 and minimizing water loss. The C4 plants are twice efficient than C3 plants in terms of fixing carbon for making of sugars or carbohydrates. C4 plants it lost only half amount of water compared to C3 plants to fix same amount of carbon dioxide during photosynthesis. So these are the adaptations to overcome transpiration mechanism in plants to give more yield. Thanks for watching.